So I would like to welcome you to the RNA Innovation Seminar Series here from the University of Michigan. My name is Mats Ljungman, and I'm the co-director of the Center for RNA Biomedicine here at the University of Michigan. And I have the great privilege to be moderating today's seminar with two very innovative investigators here from the University of Michigan. And I'm sure you've heard a lot of buzz around CRISPR to edit and cure a lot of diseases. And you probably heard the buzz around spatial transcriptomic. And lucky for us, both of these hot topics will be presented today by these two very innovative investigators from Michigan. So first up is Zhonggang Hou. He's a research investigator from the Department of Biological Chemistry. Zhonggang started his academic training at Peking University. He came to University of Wisconsin in Madison, where he did his Ph.D. He studied transcriptional silencing in yeast. He stayed at Wisconsin, did postdoc, and learned about embryonic stem cells, pluripotent stem cells, and how to use CRISPR to manipulate these types of cells. And then in 2015, he went to Harvard, where he was the director of the gene editing core there. And then in 2017, he came to Michigan as a research investigator with Yan Zhang's group at the Department of Biological Chemistry. And the title for Zhonggang's talk is Harnessing Diverse Compact CRISPR-Cas3 for Long-Range Genome Engineering. And if you do have questions during the talk, please use the Q&A button and write in your question, and I will moderate that in the end. So with that, go ahead, Zhonggang Hou.
Thank you, Matt, for the nice introduction. I would also like to thank the organizers of this RNA Innovation Seminar Series for providing us with this nice platform to share our work with the broader scientific community. As you can see from my title that I'm going to be talking about, or as Matt mentioned, I'm going to be talking about CRISPR genome engineering. So whenever I mention CRISPR genome engineering, most people think about CRISPR-Cas9. And understandably so, let me see if I can, okay. Understandably so, since its introduction in the late 2012, CRISPR-Cas9 definitely caused a revolution in the field of genetic engineering, as evident by this explosion of publications using Cas9 for gene editing. And then since then, Cas9 and its close relative Cas12 and Cas13 has been developed into various tools for gene editing, epigenetic control, chromatin imaging, base editing, RNA targeting, and most recently, COVID detection. And so it's definitely not an overstatement to say that CRISPR has touched pretty much every aspect of our lives. And that also led to the Nobel Prize for Jennifer Doudna and Emmanuel Charpentier last year for the Nobel Prize in Chemistry. So CRISPR is a very, very diverse system. It can be divided into two classes, class 1 and class 2. So as you can see in this slide, you can probably tell all the well-known members of the gene editing field, Cas9, Cas12, Cas13, they all belong to this class 2 CRISPR system. So member in this class uses a single protein for the target recognition and destruction, while members in class 1 uses multi-subunit protein complexes for the same purpose. So most people probably don't know that class 2 only accounts for roughly 10% of all identified CRISPR systems. So to put in perspective, it's more like this. That's a very tiny bit of the CRISPR system. And so while the majority of the identified CRISPR system belong to class 1. And so think about that 10% of the CRISPR already revolutionized the whole field. Imagine what 90% can do. And so within this class 1, type 1 system is the most dominant system in CRISPR. So it accounts for roughly 50% of the whole CRISPR system. So we really want to just take advantage of this diversity and see if we can actually develop novel tools for gene editing. So what's different between type 1 and then the famous Cas9? So here shown on the left is how Cas9 works. Cas9 uses a single guide RNA and to find its target on the DNA, then initiate double-strand break. While in the type 1 system, the target search is actually carried out by a protein complex called cascade. It uses the CRISPR RNA to find its target on the DNA. Then once it finds this target, it recruits a separate protein called Cas3. Cas3 has both nuclease and helicase activity. And then so once it gets recruited to the target, it travels along the DNA and then cleaves DNA along the way. The end result is large fragment DNA deletion. And so we really wanted to target this cascade Cas3 to human genome and see what it can do to the human genome. And so this is a very nice collaboration between our lab and Ailong Ke's lab at Cornell University. Ailong is an expert in the type 1 CRISPR structure and biochemistry. And so one of the reasons the type 1 system is so underutilized is the nature of its composition. So we have to express 1, 2, 3, 4, 5, 6 protein coding genes plus a CRISPR for it to function. And so that's probably why many people prefer to use a class 2 system. And so just to increase our chances of success, we decided to use the type 1E CRISPR from T-Fusca. This is a system that Ailong studies and knows a lot about this system. And then our strategy is actually to first purify the cascade complex. Instead of expressing in each individual subunit in the human cells, we actually first purify the complex already loaded with the guide RNA from E. coli, and then we purify Cas3 from E. coli separately. And this is done by a then graduate student, Adam Dolan, in Ailong's lab. Then once we get the purified protein, we electroporate the protein into our reporter cells. The reporter cells we use is a human embryonic stem cell that's labeled with a TD tomato and GFP, so it's a dual reporter cell line. And so then we would deliver into the reporter cell line, and then we design 2 guides. One targets GFP, the other targets TD tomato. And so we were very happy in our first try, we got some positive result. So as you can see in this facts plot, on the very right, this is the when we deliver the GFP targeting cascade and plus Cas3 protein, we started to see a GFP negative population. Well, if you leave out cascade or if you leave out Cas3, the GFP population is not there. And also if you use a non-targeting CRISPR, and then that also has no effect on the GFP expression. And this activity is also programmable. And so if we load the cascade with the TD tomato targeting guide, then instead of GFP negative cells, we start to see TD tomato negative cells, which means that this is basically a programmable DNA targeting module. Since, as I mentioned before, type 1 introduces long DNA fragment deletions. So we really wanted to see what kind of lesions that we have in these negative populations. So we designed a few primers, and then that will amplify a long fragment around the target site. So now you can see from each. So shown here is 2 amplifications. The first lane is no transfection, and the last lane, lane 4, is the sorted GFP negative cells. It's a population of GFP negative cells. So you can see that we're actually getting a ladder of small DNA fragments, which indicates that we are actually getting DNA deletions, which is also long. Some of the deletions are pretty long. Then we use the traditional Sanger sequencing and also deep sequence, next generation deep sequencing to further characterize those lesions. Shown on the slides on the left, these are each line is a unique deletion events, and the black line indicates the deleted DNA region. So the the dotted, the blue line is where the guide binds, where the guide recognizes. And so we can see that the deletion is quite heterogeneous. And so this deletion starts roughly around several hundred base pairs from the target site, and the deletion ends like few kb, tens of kb, even hundreds of kb away from the recognition site. And it's mostly unidirectional. So it only happens on one side of the guide RNA. And then use deep sequencing, we actually look at the overall deletion profile. We can see the deletion efficiency is quite uniform within the first 10 kb, and then it gradually drops. And then we can even get deletions that's sometimes up to 100 kb long. So these are basically long-range, unidirectional, heterogeneous deletions that we can get from the type 1 system. So just to see what can you actually do with these kind of systems. So first of all, you can delete a large genomic region. And so this is actually good for full gene removal. So most people use Cas9 to do gene knockouts. Cas9 introduces indels, which causes premature stop codons. So one study, a couple studies actually found that up to a third of those mutants actually accumulate the barren protein products, which will actually complicate the final readouts. But using type 1, we can probably achieve full gene removal, which will give you a cleaner screening result. And also we can use that for screen for non-coding elements. So one guide RNA actually give you a huge library of deletion mutants, which is not bad. That actually reduces your cost of screening. Instead of using Cas9, you have to design thousands of paired guides to actually achieve the same thing. Lastly, if we knock out Cas3's nuclease activity and still leave its helicase activity intact, while it travels along the DNA, it can serve as a long-range delivery platform for, say, for epigenome modifier or for other modification enzymes. So after we publish our results, and several other labs also publish results using type 1 for either gene editing or gene activation or transcription modulation in eukaryotes. And not coincidentally, everybody uses a type 1E system. Type 1E is actually the most, it's probably because it's the most well-studied system within type 1. However, it's also very big. It's roughly, the whole system is roughly 7 to 8 kb long. And then, so that makes the delivery via DNA or virus a bit challenging. And then while protein purification is also not easy for everyone. So just to make this tool more friendly to the broader scientific community, we start to look for the more compact systems and smaller systems. Since type 1 is so diverse, then it's probably easy for us to find one. And then we focused on the 1C system, which is small. It's the most compact system in type 1, and it's roughly 5 to 6 kb long. And then so it has only 3 genes for cascade, so Cas5, 8, and Cas7, and plus a Cas3 for DNA degradation. And so a former master student, Ryan Kruger, used bioinformatics and found one 1C system in our favorite bug, Neisseria, and it's from Neisseria lactamica. It has this CRISPR system. It has a very nice 1C system, and then he used the bioinformatics to define the PAM sequence. Then he cloned all the required genes into E. coli, and then he showed that in E. coli, this system is functional. It's basically very active through plasmid interference. So once you induce the induction of the CRISPR system, you see all the E. coli start to die off because they lost the plasmid for resistance. Then another master student, Max, purified the protein. So we purified the cascade complex. Then we also purified the Cas3 protein. We delivered into the same reporter line. And this time we were very pleasantly surprised that this system is highly active. And so we're getting roughly 50% of the GFP negative cells in using the same reporter, while we're getting roughly 10 at most to 15% with the previous system. And so this is all very nice, but protein purification is not that easy. And then just to make make this more user-friendly, we decided to develop a DNA-based delivery platform. And so we cloned all the cascade encoding genes and Class 3 into an individual plasmid for human cell expression. and then also a CRISPR expression plasmid. We transfect the cells into our reporter line, and then we were kind of disappointed to see that the efficiency is quite low. So pretty much very close to background. And while Cas9 gives really high efficiency in terms of GFP inactivation. And we check on the protein expression, all subunit expressors. And so what's causing this? We're really not sure. There's a million different reasons that might lead to this low efficiency of plasmid-based editing. And so we were stuck. Then came in a new PhD candidate student, Renke Town. And so it's all nice to have a fresh set of eyes to look at your result. And so she looked at the maxis purification and then of the cascade complex and noticed that this was banned. This was a band that we all basically dismissed as a degradation during the purification. But she actually cut out the band and sent for Edmund N-terminal sequencing. And this band actually matches to the C-terminus of Cas8, one of the subunits for the cascade, basically this one. And so it matches to the C-terminus. It starts with, actually has a start codon. It also has a predicted ribosome binding site, which indicates this is probably internal translation, alternative internal translation product. And then Renke mutated the ribosome binding site and also the start site. And indeed, that band is gone. Indicate that this is indeed a internal translation product. Then when she knocked out the Cas11, then she tried the purification again, see what happens. And so what she found that once you knock out Cas11, the complex basically falls apart. And so you're shown here on the left, you're looking at the gel filtration column. And so the black line is basically wild type purification. So you can see a very nice peak showing the cascade complex formation. And then the dotted line showing Cas11 knockout expression. So you don't actually see the cascade complex form at all. And this complex formation can be rescued by supplying Cas11 on a separate plasmid. And so by showing here on the orange line, you see a nice peak showing the cascade. And then, so she was able to purify both the wild type and the Cas11 rescue RNP when we deliver to the cells, and both of them are pretty active. And so maybe that's the thing we're looking for. That's the discrepancy between RNP and plasmid-based editing, because the internal translation signal for bacteria cannot be recognized by human cells. So in human cells, there will be no Cas11. Just to test that hypothesis, Renke basically cloned Cas11 onto its individual plasmid and then transfected all these plasmid into human cells. And then we were very happy to see that completely rescued the plasmid-based targeting. And so you can see without Cas11, then we get really low level of editing, while when you add in one Cas11 plasmid, the editing efficiency goes up 10 to 20 fold. And so this is very nice. And also the same strategy, but basically putting Cas11 in there can actually enable also mRNA-based editing. And so we can express all these guys as mRNAs, and then we can electroporate the thing into our reporter cells at pretty high efficiency. So we can use RNA for CRISPR, then we can also use DNA plasmid for CRISPR, and then we get higher efficiency if we use plasmid to express CRISPR. And so we believe the supplying Cas11 really enables very robust editing with plasmid or mRNA. And then at the same time, the other labs also identified this internal translation product in type 1b and type 1d and other type 1c system, indicating that this is probably a universal phenomena for the type 1 system, for those compact systems. 1b, 1c, and 1d, they're all very compact systems. So there's a universal phenomenon for all these compact systems. And so indeed, we try basically using this strategy and to see if we can get other systems, other 1C or 1D or 1B system to work. And then putting Cas11 in there definitely makes them work much better. So here's a different 1C system. And so without Cas11, you get pretty much zero editing, but once you put in a Cas11, that increases to roughly 20%. And then for this 1D and 1B, they also Cas11 increases the editing efficiency for the whole plasmid-based delivery by a lot. And so we believe that this using Cas11 actually opens the door to utilize a lot of compact type 1 systems for gene editing or for other for purposes. So I guess that's all I have for today. And then I would like to thank, so this is definitely a team effort. I would like to thank all the members in the Yan Zhang's lab, especially Yan, for starting this very bold project. This is a high-risk project when she just started her lab. And then also Max and Ryan, Rinke, they all putting lots of effort into this project. And I would like to thank our collaborator, Ailong, and then his grad student, Adam, for purifying those proteins, Yibei and Chunyi for all helpful discussions. And then last but not the least, and Peter for very nice support for bioinformatic analysis. Thank you, and I would like to take questions.
Thank you so much. That was a very interesting talk. Please put in questions under Q&A. I can start with one question. So if you were to compare the toxicity of inducing a double-strand break with Cas9 versus doing the shredding with Cas3, what would you say? Is it a lot more toxic to do the shredding versus a double-strand break?
We actually did not. So based on where you target though, and then if we target non-essential genes and say GFP reporter, we actually do not really see high level of toxicity associated with the delivery of our cascade or type 1 CRISPR. And then most of the toxicity we actually we actually have is as through the plasmid transfection. The plasmid itself, DNA is kind of toxic to the cell. But if we switch to RNP delivery, that's actually not too bad.
Okay. Okay, we got a question here. Let me read it for you. This is from Catherine Skoll. Great talk. Do you have any thoughts on what drives the heterogeneity of the length of the deleted regions?
That's a very, very good question. And so there's lots of work been done in vitro by single molecule just to look at how Cas3 or cascade versus plus Cas3 introduces DNA deletions. And then it's not completely clear yet. And so the idea would be that the lens is probably affected by local chromatin structure. And so there's a certain chance that when Cas3 hits certain structure, it will just fall off. And there's a certain chance it will actually plow through that secondary structure or other blocks. And so it's basically by chance it will just fall off at a certain point, and then other batches will just go on, eventually falls off. So that's probably one of the reasons that the deletion is quite heterogeneous.
Okay, please. Oh, here's another one. Jifeng Zhang, how about the off-target effects of this Cas3?
Yes, so Cas3 itself will not actually degrade DNA, a double-stranded DNA. So if you just mix Cas3 with double-stranded DNA, it does not have any activity. So only when it gets recruited by cascade to the target site, then it starts to travel and degrade DNA. So just Cas3 itself does not have off-target effect. And then off-target effect mostly happens with the cascade. And then we did some off-target profiling, and then we found that it tolerates it's kind of like a Cas9 within the first few bases of the guide RNA. Then it's very sensitive to mismatches. Then outside of that, it starts to tolerate mismatches. And then so we're still working on the off-target profiling at this moment.
Mm-hmm. Another question I have. So could you swap out the Cas3 shredding activity and place that on Cas9. So you would have an RNA targeting module from Cas9 hooked up with Cas3, so you get a shredder. So just kind of circumventing the cascade proteins and just.
That's a very interesting thought. We never thought about it. Yeah, sure, absolutely. That we can definitely give that a try.
Okay, sounds good. I mean, we're working with a dead Cas9 that has a endonuclease attached to it. So I think there could be a lot of interesting things you can do with adding various things to, you know, swapping domains. And in the end, it's to find something that's small and efficient and so forth. And in terms of using the Cas3 system for gene editing, what are the types of diseases you think it will lend itself very well for?
So basically we're looking at, say, if you have certain cancer genes that you want to get rid of, that's one thing. And then if you want to screen for non-coding, so there are non-coding regions that will cause diseases, and single point mutation or small indels that might not get rid of that non-coding sequence, then using type 1 will probably be a better choice. And then also in terms of screening, this would be a very nice screening tool that you target this guy to a certain region, then it will just randomly create deletions of varying length. Then you can screen for non-coding elements, the important elements within that region, using that library or that pool of deletions.
Very interesting. So the Cas3 is both the helicase and an endonuclease?
Helicase, and then I believe it's endonuclease. Yes, that's correct.
Okay. Are there any other, like in the mammalian genome, any other genes like that has that dual function of being a helicase and an endonuclease, or?
That's a good question. I'm not aware of anything, but that doesn't mean it doesn't exist.
Right. That's very cool. Okay, any more questions from anybody? Last call here before we switch over. If not, thank you again very much, Zhonggang, for a very interesting seminar. And please go and pick up your, was it son at the?
Yes.
Yes. Don't forget that. Go ahead and do that. Thank you again.
Thank you.
Okay, so we are good on time, and let's switch over for the second talk by Jun Hee Lee. Jun Hee is an associate professor at the Department of Molecular and Integrative Physiology here at Michigan. He started his academic career at the Korean Advanced Institute for Science and Technology, where he did his undergraduate. He did his PhD and his first postdoc there. Then he got a chance to go to University of California at San Diego, and he did his second postdoc there before coming to Michigan a decade ago to the Department of Molecular and Integrative Physiology. And Jun Lee, now I don't have your title in front of me, but I'm sure that's on your slide, but you're going to talk about a very innovative and new technology to look at spatial transcriptomic that you and your group developed. That's called Seq-Scope, and it was just published in Cell. It was a really, really nice study. So congratulations on that. And we look forward to hear more of the details from you now. So go ahead.
Thank you, Dr. Ljungman, for nice introduction. And I really appreciate that I have this opportunity to share our recent technology named Seq-Scope with you. So yeah, before we talk about the Seq-Scope technology, I wanted to briefly remind you about the importance of a microscope. So you know that microscope has been very essential for the development of biology, because it can allow us to see very, very tiny structures which cannot be observed through our naked eye. So it can show cellular, subcellular structures, and it has been very essential for development of microbiology, histology, many important biological disciplines. And now we are living in molecular biology era. So basically this microscopy has been combined with molecular detection techniques, including immunohistochemistry, which can detect proteins, or RNA in situ hybridization, which can detect different RNA sequences. So we can now see specific biomolecules from the histology section, but those methodologies are actually limited because in a single session of histology, they can only monitor a limited number of proteins or RNA species. In contrast, an emerging technology named spatial transcriptomics aims to examine all the genes transcribed from the genome at once from a single histology slide. So typically, spatial transcriptomics is implemented using specially barcoded oligonucleotide arrays. So in those arrays, we can find RNA capturing molecules like oligo-DT shown here. So they can capture mRNA molecules. And those RNA capturing molecules are encoded with a special barcode sequence. So a specific nucleotide barcode sequence can encode the spot XY coordinates. So once we overlay the tissue section onto this array, and then RNAs will be released from the tissue, and then these RNA capturing molecules will capture the RNA, and then we can perform conventional reverse transcription. So the result is actually the chimeric library that has both special barcode sequence and cDNA sequence. And then we can sequence this library, which is actually the RNA-seq. So we can reveal the gene, and using the special barcode, we can actually pinpoint where those specific gene expression is originated from the tissue slide. So this special transcriptomics has been now already implemented in a commercial solution. And 10x Genomics last year launched the 10x Vision, which is the first generation spatial transcriptomics. So this special transcriptomics solution actually was very good in profiling, especially profiling the gene expression landscape. But it has also a limitation because its spot-to-spot, center-to-center resolution was limited at 100 micrometer. So actually, if we compare that resolution with our naked eye, actually that resolution was worse than our naked eye. So actually examining the histology slide using Vision was almost similar to examining the histology slide with your own naked eye, probably at 1 meter away. So even though it can show some special features, actually it didn't reveal the details of tissue histology. So people has been aware of this problem, and then there has been a lot of recent developments which used either microbead or microfluidics devices so that we can actually decrease the center-to-center distance, increasing the resolution. Indeed, these methods, even though not commercialized yet, they can improve the resolution down to the 10 to 20 micrometer resolution. So this has been an order of magnitude improvement, but still their resolution is around the level of 2x magnifier, which can actually not see the microscopic structures. So here is the problem. So to examine the microscopic image details, so this image is hypothetical 1 micrometer resolution image. But if you look at those structures in either a magnifier or a naked eye, actually those special details are all obscured and unobservable. So this has been a big problem. And we imagine that if we are able to generate a very, very tight arrangement of spatial barcode, which is, let's say, comparable to the microscopic resolution, then we can perform microscopic spatial transcriptomics. And then if we are able to really implement that, probably it may have a potential to replace a lot of different iterative immunostaining or RNA in situ sessions, saving a lot of time and money. And also it can allow for special single cell analysis because this microscopic resolution will allow single cell examination. And also it can allow for the subcellular analysis because it can show the microscopic structures. So we were able to provide such a solution called as Seq-Scope or Seq-Scope. So here we were able to make a very, very tiny arrangement of differently labeled spatial barcode. So if you compare those pixel structures with a single pixel BGM area, you will be able to see a day and night difference. So this Seq-Scope is actually initiated by generation of single-stranded DNA oligonucleotide library. So this library will have PCR handles, RNA capture sequences, but importantly, it will have a random nucleotide, which we call as high definition map coordinate identifier, or HDMI sequences. In this oligonucleotide library, theoretically, each of the library components will have different structures because it will have a lot of sequence diversity. So each of the single molecules will have different sequences, and then we amplify them on Illumina flow cell platform, where it has PCR adapters coated on the surface. So basically, we can perform the two-dimensional PCR, which can generate different HDMI clusters. And then using Illumina sequencing by synthesis method, we are able to determine the sequence of each cluster and their XY coordinates in the spatial space. So since this is a PCR reaction, each of the cluster has unique sequences, but all the molecules within each cluster will have identical sequences because they are all the daughter molecules of initial HDMI oligo. And then we process process each of the clusters so that it can expose the RNA capture moiety, and then we overlay our tissue sections onto this Seq-Scope array, and then using after that we are able to generate another library that is a chimeric molecule between the HDMI sequences and the cDNA sequences. So we call the first sequencing process determining the XY coordinate as first seq, and the RNA-seq process as the second seq. So Seq-Scope is composed of the first seq and the second seq process. So first seq of the Seq-Scope will generate a data table that has spot XY coordinate and at the same time HDMI sequences. So basically in the first seq, you will have each spot XY coordinate and HDMI sequences determined. And second seq is basically an RNA-seq. So you know about the gene expression, so you can get digital gene expression metrics. And at the same time, each of the gene expression is associated with the HDMI sequence. So basically using the HDMI as a spatial index, you can relate each gene with a coordinate. So each of the gene expression counts from the RNA-seq second seq experiment can be associated with the XY coordinates that were determined through the first seq process. So theoretically, because we determined that the spot-to-spot distance is just 0.6 micrometer, theoretically we can do the microscopic examination of spatial transcriptomics. And then we wanted to know really it can support the microscopic analysis and whether we can really see the subcellular structures from the cell. Now, you know that all the RNAs from the cell are basically transcribed from the nucleus as an unspliced messenger RNA. But before they are transported into the cytoplasm, they need to be spliced. And then some of the RNAs enter the mitochondria, but mitochondria has its own genome, so it has mitochondrial encoded RNA. Because Seq-Scope is basically an RNA-seq, we have all the information from the RNA sequences. And then using the sequences, we can know whether that sequence is either unspliced, spliced, or mitochondrial. So we actually plotted unspliced, spliced, and mitochondrial encoded RNA sequences onto a histological space. And we were able to see that actually we can see a structure that resembles nuclear cytoplasmic structure. We can see that unspliced RNAs are confined in the nuclear-like region, and spliced and mitochondrial RNAs are dispersed throughout the cytoplasm microregion. Even when we randomly divided the sequences into the 3 different pools, those observations were reproducibly observed. And we can even do some subcellular analysis so we can identify all the nuclear-like regions. And then we can isolate mRNAs that are enriched inside the nucleus. And we can have a list of genes that are nuclear-enriched genes. And actually, there is a recent study which performed physical separation of cytoplasm and nucleus. And then they performed the differential expression between the nuclear expression of mRNAs and cytoplasmic expression of mRNAs. And they showed that a class of mRNAs are enriched in liver cell nucleus. And this is the liver tissue. And actually, we can see that the top genes isolated through the physical separation experiment are consistent with our bioinformatic nuclear identification experiment performed using the Seq-Scope dataset. So basically, their gene lists were almost identical, meaning that Seq-Scope can indeed enable spatial subcellular transcriptome analysis. Now we wanted to make sure it really represents the nuclear area, that unspliced RNAs are really representing the nuclear area. So we overlaid them with hematoxylin and eosin staining, and you will be able to see that really the nuclear region shows high expression of unspliced RNA. And using computational cell segmentation method, we were able to even isolate the single hepatocellular area. And using those coordinate information, we can collapse all over the RNA sequence into the single cells. And then actually the RNA information isolated from those single cell area is actually comparable to conventional single cell analysis technologies like DropSeq or MarsSeq. And we can also perform the conventional single cell analysis as we performed the other single cell data sets. But the unique point of Seq-Scope is that actually they can perform the spatial single cell analysis. So basically you can identify each spot originating spatial space. So you can plot them onto the spatial space, and you can even overlay them with histological structure. And you will be able to see that actually those spatial classification is consistent with histological findings. So around the portal vein region, we were able to find periportal hepatocyte phenotypes, and around the central vein region, we will be able to identify pericentral hepatocytes. Furthermore, Seq-Scope can also enable examination of individual gene expression. So basically, like a conventional gene search engine, we can enter the individual gene name, and actually Seq-Scope can monitor microscopic gene expression patterns for the whole transcriptome. So for instance, pericentral genes like GLUL is only expressed in central area, while the periportal genes like albumin and MUP20 can be found from the periportal region. So such details in microscopic gene expression was only observed in Seq-Scope dataset. And when similar analysis was done using former spatial transcriptomics methodologies, actually such type of fine microscopic details were not observed. And those big differences are basically because of big quantitative differences. So Seq-Scope was able to provide very high resolution down to 0.5 to 0.7 micrometer. And also it can reveal many transcripts per area. So those types of quantitative differences led to such a big difference in image quality. I only showed the hepatocellular expression, but the same data set also revealed the hepatocyte expressions, such as macrophage, red blood cell, and endothelial cell, and hepatic stellate cell transcriptome from the unbiased segmentation analysis. And we were able to locate those individual cell types as well. But the important thing is those non-hepatocellular cell populations are dramatically increased when livers undergo injury and fibrosis. So we are able to isolate many different non-hepatocyte cell types from injured liver samples. And indeed, those new cell types are really associated with the liver injury and fibrosis. And we were also able to examine them in a microscopic scale, and we can also overlay them with histology, so we can associate specific pathology with the transcriptome phenotypes. Because it has so much information, I just wanted to show you a simplified diagram. So in this example, you can show that dead hepatocytes are surrounded by inflamed macrophages, and then they are surrounded by injured hepatocytes, and then normal hepatocytes and macrophages are far away from those injury sites. And same finding was observed through immunoconfocal microscope. And also similar findings were found from the fibrosis site, where activated hepatic stellate cells and inflamed macrophages are closely intermingled with each other, which is again confirmed through immunofluorescence microscope. So basically, I just showed the liver data, but the similar findings were also made from the colon. We can identify different cellular layers and also individual cell components. For instance, in the colonic epithelia, we can find many different epithelial cell types, including stem cells and surface colonocyte and many other cell types performing different actions. And also we can identify many different non-epithelial cell types, including macrophages, B cells, entering neurons, and smooth muscles. And we were able to also identify the microscopic structures of all those cell type arrangements. And those arrangements were consistent with what we observed from hematoxylin eosin staining of the same section. Basically, we were able to see that there is a boundary between epithelia and lamina propria. So epithelia and lamina propria. So epithelial cells and non-epithelial cells are only found in their corresponding histological locations. And also we can find the cell boundaries as well. And also we were able to identify many different cell type specific marker genes, and how we can also examine how those cell type specific markers are expressed in the spatial space. So because of the time, I will not go into the details of other assays, but Seq-Scope can also examine some tissues like skeletal muscle, which has elongated myofibers that are not amenable for the typical single cell assays. But we were able to identify their individual myofiber types and their arrangements, and also many other subcellular features and other cell types. And especially, we can also identify neuromuscular junction signature, which are only observed in their restricted anatomical locations. So these details were not approachable through formal spatial transcriptomics technology. Also, we were able to approach the skin diseases and the disease cell types associated with skin diseases like acne. So we can identify inflamed keratinocytes and their associated macrophages and fibroblasts. And also we are able to associate them with a specific histological structure at the microscopic scale. So let me briefly summarize what our new Seq-Scope technology can offer in future. So typically, bulk RNA-seq is often compared to making a fruit smoothie made out of a nice fruit tray. Even though this can show the very deep analysis, actually you destroy all the spatial information as well as single cell information. So single cell transcriptomics, such as single cell RNA-seq, you can individually examine the single cell transcriptome, so you can identify different cell types and their transcriptomic contents, but actually you still lose the spatial information of how the original tissue has such an arrangement. This can be addressed by spatial transcriptomics, but so far the resolution of spatial transcriptomics are not enough to identify single cells. So basically you are making just a number of different smoothies that has a spatial location. And now we are improving this with multiple different technologies, so we can have a near cellular resolution. But Seq-Scope can further advance this through the field, and then actually we can have a real single cell resolution in examining the spatial transcriptome. And with its ultra high resolution, actually, not only the single cell information, but also the subcellular information can be obtained through the Seq-Scope. So we are very excited to apply this to multiple different tissues. So now within our department, in our university, and several other collaborators outside of the university, we are looking into applying this Seq-Scope experiment into diverse organs. So I wanted to finish my talk here, and especially I wanted to thank Dr. Jun Cho, who implemented all the complicated experimental procedures, Jin Yu, and our collaborators, Dr. Hyun-Min Kang and Gu Jun, for making a nice computational pipeline for analyzing these data sets, and also our collaborators who provided tissues and helped us with data interpretation. So thank you very much and for your attention, and I would like to take any questions.
Well, thank you. Thank you so much, Jun Hee. That was really amazing. So I'm sure that you have a lot of people knocking on your door want to collaborate on all their different special tissues, right? Do you have it set up for like a core facility or something like that, or is that in the plans?
Yeah, so that's certainly a plan for making this more accessible. So actually, this technology is still new. It has not passed 1 year after we actually have our first data, and then we have worked diligently on kind of optimizing and scaling up. So our initial data set was produced in very tiny 1 millimeter circles, and then we wanted to kind of expand the area so that we can analyze more tissues and with less cost. And we are still making progress, and we are open to collaborations and testing the tissues. But so far, the challenges would be mostly on the tissue quality and the tissue optimization protocols. So those things still needs to be optimized, and I hope that we are able to update some that so that we can have a more defined pipeline of analyzing the tissues and also scale up. So it's definitely our interest of providing this more as a service, so that it can be accessible by many different researchers.
Yeah, that would be a great resource. Do you think that you're sort of hit the end of what you can do with resolution, or do you think there is a layer beyond what you've done that you can go even more higher resolution?
So I think we almost hit the boundary of what we can do with this concept of methodology, because this concept of methodology is depending on the nucleic acid diffusion and capture. And then since they will be diffused, we estimated the diffusion rate as around the diameter of 1 to 2 micrometers. So I think this is some micrometer resolution, even though, those are kind of will show the normal distribution. So I think we are almost at the limit. And also at the data depth, it's also very approaching the limit. And we think that we probably capture almost everything that are present within, let's say, a couple of micrometer thickness section. But since those are very narrow section, so probably what you will observe is, single plane examination from, let's say, confocal microscopy. So probably you will not discover a lot of transcription there unless you can combine those. And another limitation that we wanted to address is, of course, the cost, because actually the array cost itself is very, we have bring down very cheap levels. So for instance, you can examine one tissue, let's say with a couple of $100 array, but the problem is the sequencing. So you need to sequence a lot to get a difference. For instance, typical RNA sequencing will produce around 20 million reads. But if you divide that with, let's say, 1 square millimeter imaging area, you don't have that much depth. And especially for rarely expressed genes, probably you will not get that much count. So to kind of address those, we are using different approaches in targeting or protein signature. So we are still working on improving those aspects as well.
That's great. So a question that connects with what you just said. Jifeng Zhang is asking, in general, how many genes can you catch in each cell? So the initial,
our initial data set, actually, we spent quite a lot of money into sequencing the samples. And then actually our estimation says that we only examined, let's say, around 50 to 60% of total library complexity, but still we can capture around 22 transcripts per micrometer, which means that if it is 10 micrometer square, then it will be around 2,200 unique transcripts. And like big cells, like hepatocytes, they will have, let's say, up to 50 micrometer or 30 micrometer diameter, and then it will be much more. So it depends on the cell type, it depends on the area of tissue, and also importantly, how deep you want to characterize. So yeah, to get 22 UMI per micrometer, you need to sequence really, really deep. So yeah, those are some kind of practical considerations of how much information you wanted to get. So yeah, that depends on the application. So if you already have a single cell data set, you wanted to map them into the spatial space, or you want to kind of construct de novo cell type mapping using the Seq-Scope. So that all depends on the application, I guess.
Okay, we have another technical question here. Can you explain how to aggregate UMIs across different HDMIs to obtain single cell level gene expression?
So far, what we have done is we perform the image-based cell segmentation. So from the, because before we perform Seq-Scope analysis, we perform the, typical histology image staining like hematoxylin and eosin. And then we marked up cell boundaries and we collapse all the, coordinates within that boundary into the single cell transcriptome. That's what we did for liver data set. And for colon data set, we used a different colon and other data set, we actually used a different approach. So basically we do random gridding of, let's say, 10 micrometer square grid. And then after performing cell type mapping, actually we kind of refined those grids so that we can estimate the cell boundaries. So we use the sliding windows method so that we can oversample the initial data set so that we can identify the boundaries between different cell types. So those are two different methods that we have implemented. But of course, there may be another method of detecting the virtual cell boundaries just based on the data set. So those methodology, we would look forward to further development in those methodology area.
I don't see any more. I have a question. So you showed some pictures where you had looked at spliced versus unspliced RNA. What do you look for in the unspliced population? Is it just intronic regions, or what are you looking at when you show those pictures of the nuclear?
Yeah. So for that examination, actually we used velocity analysis. So those analyses were actually done in single cell area to know what genes are being transcribed and what genes are being expressed so that they can have temporal resolution. But we just utilized them for plotting unspliced and spliced ones. So basically they detect, I believe, the spliced junctions, and then you see whether those are really unspliced region. For instance, you can see the intron, or you detect the exon region. So and for the spliced region detection, probably we wanted to have those spliced junctions. So only spliced transcripts will show such sequence.
Okay, and then you had another picture of, you showed like a tissue with lots of cells in them, and then there was a few cells that had a really dark nucleus for unspliced.
Yep.
But then there was a lot of other cells that didn't have that. Could you interpret that to mean that certain cells are really poor at splicing while other cells splice really well?
Yeah, so those are really interesting observations. So actually, we were initially puzzled over why some nucleus are showing the strong signal, but while the others are not. And actually, that's because the tissue we overlay onto our spatial array is quite thick. So those are 10 micrometer thick. And effectively, our methodology will only capture the lower, let's say 2 to 3 micrometer. And then in those sections, some of the nucleus will be placed here, some of will be here, but some of the nucleus will be placed downwards. And then we think that that's probably there will be some variations in unspliced, unspliced gene expression, but it is also possible that they are spatially, distributed. So some nucleus may be very strong in H&E, but they are not very well represented in our data set. But some nucleus will be very, very weak in H&E, but they will be highly represented into our data set because they are more efficiently captured. Yeah, so we think that both are possible. So there may be some internal variations, but it is also possible that it is just because of their spatial arrangement.
Yeah, there's so much to look at, right? I mean, it's so exciting to be able to look at things like this, and there's probably more questions than answers that you stumble upon when you look at it.
Still we are learning about the techniques, and especially as we collaborate with multiple people. Actually, for some of the projects, it's very challenging. For some, actually, we didn't expect, but it shows the data. So actually, we are still learning about how the processes are done. So hopefully, this develops further and it can make good contributions.
Yeah, it's very, very exciting. So I want to thank you, Jun Hee, and also Zhonggang before for great presentations. And I thank everybody for their attendance. Ailong Key is giving a seminar tomorrow at noon, and he was mentioned earlier today about the CRISPR. So that's at noon tomorrow, so you can tune into that. So with that, thank you so much from University of Michigan. Have a great day. Thank you.